Thank you to the organizers for providing me the chance to speak today. My name is Amy Wesolowski, and I'm an assistant professor in the epidemiology department at Hopkins. Today I'm going to speak about seasonal human movement and the consequences for infectious disease transmission. Human travel can result in pathogens being introduced into susceptible populations. Here are 4 historic examples of how human travel has led to the global spread of various infectious diseases, all the way back to the plague throughout Europe, the Middle East, and North Africa, starting in the 2nd century, to the spread of SARS-CoV-1 from international travel in the early 2000s. What all of these examples demonstrate is that humans are able to introduce pathogens into susceptible populations that can result in expansive disease spread. The current SARS-CoV-2 pandemic is yet another example of how human travel can result in rapid disease spread globally. Here's an example from the earlier stages of the pandemic where an individual was infected in Singapore who then traveled to a ski resort in France. They were then in contact with a number of other individuals, many of whom traveled again within a few days or weeks and were able to introduce the pathogen into these susceptible populations, resulting in secondary cases. Ultimately, the type of mobility we are interested in, both in terms of the temporal and spatial scales, will depend on the pathogen of interest. For example, some pathogens, like measles, we may be interested in understanding national mobility patterns. For other pathogens, like dengue in some instances, we may be interested in very fine spatial patterns of travel if we think that risk is highly stratified locally. Ultimately, the natural life history of the pathogen, ability to identify the susceptible population at risk, and understanding of how travel interacts and impacts the disease process will dictate the ideal spatial scale we would investigate. There are many different kinds of data sets available to quantify travel patterns on various temporal and spatial scales. There are a number of population-level data sets, such as census data or flight information. One of the benefits of these may be that they may be available in multiple countries and may be collected to be representative of the population of interest. There are also multiple individual-level data sets that can be used to quantify individual mobility patterns on a wide range of spatial and temporal scales. These data could also be collected for a particular population of interest and may be able to collect information about the infectious process or prophylaxis while traveling that may be relevant for disease transmission. However, like all data, these data sets often have limitations. It may be difficult to collect large samples of individual level data, census or migration data may not be collected very frequently, and flight information may only represent a small proportion of all travel that is relevant for disease transmission. These data may also have gaps in particular geographies. Here's a plot showing the population versus the number of international airports, and for example, in Africa, there are fewer airports per capita. This may impact the generalizability and utility of these data in particular settings. Ultimately, we don't really need to measure every single trip by every single person in the entire universe forever. We actually just need to understand what dimensions or aspects of travel are relevant to disease dynamics. And in particular, for seasonal aspects of travel, we need to understand how seasonal travel patterns may be impacting or driving disease transmission. Here's an example of estimates of seasonal transmission coefficients of measles. It has long been known that school terms and the aggregation of kids in schools has driven measles transmission dynamics historically in many settings. In this instance, we would really want to know how individuals are traveling seasonally and if this can be related to school terms, which can then be used to predict when and where transmissions of measles may increase. We may be interested in quantifying human mobility patterns seasonally to understand if there's times of the year when infected individuals are traveling, which may result in the disease being introduced into susceptible populations. Times of the year when behaviors such as fishing or farming may change that lead to an additional exposure event, or when there might be an influx of susceptible individuals such as migrant populations that can result in a disease outbreak. Here's a summary put together by a postdoc in my group, Hannah. Various data sets may or may not be able to capture these patterns. For example, GPS loggers may be able to capture very detailed patterns of seasonal mobility, but you would need to deploy these at the correct times of the year and keep the same individuals enrolled for longer periods to measure these patterns. Things like satellite imagery may be available at various times of the year, but you would actually need to be able to understand how these relative or proxy measures are able to translate to actual trip counts. For example, here are data collected from Zambia that a postdoc in my group, Catherine's working on. These data were collected as part of a study of malaria in an area of Zambia that has distinct dry, shown in brown, and rainy season, shown in blue. As you can see on the left-hand side, the proportion of time an individual spends at home can vary greatly between the wet and dry seasons. And on the right-hand side, these data from an individual show that the distance and direction an individual will travel will also depend on the season, with travel during the dry season going much further distances and in different directions. How it will always be difficult to collect these data for entire populations. And due to these large data gaps, we have been working with mobile phone operators to process and access population estimates of mobility from billing records. Briefly, the way these data are processed are that when an individual subscriber is making or sending calls or texts or data, we can infer their most used routing tower and location where they're spending the majority of their time per day or per hour. Using these location estimates, we then infer travel based on when an individual subscriber does or does not change a location. Here's an example of work we did a few years ago where we analyzed mobility patterns inferred from mobile phone data from 3 different countries, Kenya, Pakistan, and Namibia. We processed each data set in a similar manner to then estimate travel patterns over time. Here are the data just from Namibia. Each row in this plot is a district with northern districts on the top and southern districts on the bottom, where each color represents the percentage of trips to other districts occurring that day. These data just show the trips measured from January to December in 2013. The black line is the country average, and the first thing we can see is that there are peaks of travel between districts during school breaks, shown in dashed lines, including the Christmas holiday, shown at the end of the plot, with Christmas showing the largest peak in travel between districts. We saw less of an effect of travel during the wet and dry season. We also saw consistent patterns between years, with data from 2011 to 2013 all showing similar patterns with increases in travel during school breaks, with the largest increases often occurring in December and January. We can then also look at different patterns by routes, by different rural and urban areas, since different routes of travel may have different seasonal patterns, to see if there are were more or fewer relative to the number of expected trips of each route type over the course of the year. And once again, here are some examples from Namibia, with travel from rural to urban areas shown on the top and travel from urban areas to rural areas shown on the bottom. What we saw is that travel from rural to urban areas increased in both December, shown in brown, and January, shown in light blue, with larger relative increases in January. In contrast, urban to rural travel only had a large relative increase in December, the brown box plot. This corresponds to individuals from urban areas traveling back home for the holidays in more rural areas, and these data were able to capture these differences. Ultimately, these differences in seasonal travel will only be important for disease dynamics if they're related to differences in how the disease may spread, the likelihood of an outbreak, or who may be infected. We used a simple transmission model to look at the effects of different travel patterns in January and December to see if these changes will impact the disease transmission dynamics. What we saw is that in different areas of the country, the simulated pathogen may arrive earlier or later if you use travel that's representative of January or December trip estimates, and that these patterns will depend spatially, with some areas of the country, such as less populated southern areas of the country, arriving later in December than January. Human mobility can impact the transmission of various infectious diseases, and there are many different ways that we can quantify human travel, but many of these data sets are not actually able to capture detailed temporal information on seasonal travel. For some of these data sets, we are actually able to quantify seasonal travel patterns, such as mobile phone or GPS loggers, but their impact on disease processes will really depend on the type of pathogen, and more work should be done to validate the importance of capturing seasonal trends of travel and different ways to model these. There are a number of individuals who are involved in this work and a number of different funders. Thanks very much.